Hello and welcome everybody to my channel Lumini Bioentrance, and today we will be discussing Odisha CPET Microbiology question paper conducted in the year 2021. So coming to the first question. Bacteria that retain the crystal violet stain appear purple color under microscope are. So they are gram-positive bacteria and they stain violet due to the presence of thick layer of peptidoglycan in their cell wall, and this will retain the crystal violet stain. So the answer is option C, that is gram positive. Then next one. Organism having cell without a distinct nucleus are. So both A and C, that is bacteria and cyanobacteria, whereas fungi is a eukaryote. Then next one. Bacteria having flagella throughout its surface. It is called as peritrichous. A similar question was asked last year exam also, for conducted in the year 2020. You can refer to my question paper. I have already uploaded that. Throughout its, okay. Monotrichous is having a single flagellum. Lophotrichous is tuft of flagella at one end, and atrichous is no flagella at all. Then which of the following is absent in gram-positive bacteria? So gram-positive bacteria, the LPS, that is lipopolysaccharide membrane, yes, is absent, whereas it is present in gram-negative bacteria. Then next one. Which one of the following establishes symbiotic association with soya bean plant? So it is, they have asked specifically for soya bean plant, so it is Bradyrhizobium. Answer is option C. Which of the following is not a sexual spore? So, ascospores are asexual spores. Basidiospores are also asexual spores. So, sporangiophores, they are not, is not a sexual spore. Okay, so answer is option C, sporangiophore. It is a sexual spore. Then the commonly used vector of human genome sequencing are. So, YAC and BAC. BAC insert size is less than 300 kb and YAC has around 200 to 2000 kb. So that much DNA can be inserted into these vectors. So answer is option D. Then mesosomes are enfolding or invagination of which of the following? Of the plasma membrane, and they are involved in respiration, and they are analogous to eukaryotic mitochondria. So the answer is option A, and a question related to mesosomes was also asked just the previous year. I have uploaded the question paper, you can go through that. Then the virus that attacks bacteria are called as bacteriophages. Answer is. Then a condition in which virus enters the host's bloodstream is called as. It is called, okay, we will see one by one. Septicemia, it is clinical name for blood poisoning by bacteria. Leukemia is cancer of body's blood forming tissues. Pyemia is a type of sepsis that lead to widespread abscesses of a metastatic nature. So the condition in which virus enters the host cell bloodstream is called as viremia. Answer is option B. Which of the following infections are related to COVID-19? So SARS and MERS both are related to COVID-19. So answer is both A and B. What is the characteristic of retroviruses? RNA is the genetic material, uses reverse transcriptase enzyme. Both A and B. Yes, both A and B are characteristic features of retroviruses. Then next one. Extra chromosomal DNA molecule having autonomous replication is called. So it is called as a plasmid. Answer is option A. Then next one. A nucleic acid containing 33% A, 20% T, 21% C, 26% T is called. Now this particular nucleic acid, if it was double stranded, then the percentage of A and T will be same. But here because A will form double stranded base pair with T, and C will form double stranded base pair with G. So one of them, they have given two T values, one of them must be G, it might be a printing mistake. Now if if the number of A is not equal to number of T and number of C is not equal to number of G, and if T is present instead of U, then it is a single-stranded DNA. Answer is option B. Then next one. DNA polymerase 1 has the following function. Mark the correct one. 5' 3' polymerization. Yes. 5' 3' exonuclease. Yes. 3' 5' exonuclease. Yes. So answer is all of the above. The telomerase enzyme that solves the end replication problem in eukaryotes contain. So it contains the telomerase enzyme. It contains both RNA and proteins. Answer is option A. Then which of the following conditions arise when the level of glucose and lactose are high in lac operon? So the glucose is also high, lactose is also high, and E. coli will have a preference for glucose over lactose. So first it will take up glucose, and once the glucose has exhausted, then it will start utilizing lactose. So answer here is option A, that is glucose will be taken up first by the cells. Then which of the following partial diploids will express beta-galactosidase constitutively? So there is continuous expression of beta-galactosidase. So here operator is constitutive, so LAC-Z plus will be produced constitutively. And in the second case, repressor is not there. So LAC-Z will be produced, but here repressor is present. So this repressor will go and bind to the operator of this particular 1 minute. I will explain once more. In the first case, this is operator is constitutive. So LAC-Z plus will be continuously produced. And in the second case, if you see option B, repressor is mutated in first this genotype, whereas here the repressor is not mutated and it is wild type. So this repressor will bind to the operator of this one and it will block the synthesis of Z plus. So here also it will not be produced constitutively. Same condition here also in option C also. But in option D, LAC-Z minus itself is not there. So even though operator is constitutive, LAC-Z minus Z minus is present, that is beta-galactosidase itself is not there. So even if it is constitutive, beta-galactosidase will not be produced. So the answer here is option A. Then in protein synthesis, tRNA carrying the initiating amino acid enters in which site of the ribosome? So the initiator tRNA, it immediately occupies the P site, but all other RNAs, they will enter the A site. So the answer here is option B. In prokaryotes, which subunit of the ribosome has peptidyl transferase activity? So it is the 50S subunit. Answer is option A. Mutation which do not cause any functional change in the protein are known as silent mutation. Here the codon for for alanine is replaced by another codon coding for alanine itself. So even if you changed the nucleotide from the DNA, still alanine is produced there. So it is a type of silent mutation. There is no change in the protein sequence. Then next one. Which one of the following repairs thymine dimers? AP endonuclease, MUTS, MUTL, DNA photolyase. It is DNA photolyase. Answer is C. Which of the following is not a post-transcriptional modification? So it is glycosylation. Answer is option D. And then next one. RNA polymerase binds to which site or region of the DNA molecule? So RNA polymerase, it binds to the promoter. Operator is bound by the repressor. Okay. Then next one. Which of the following organism is widely used as biocontrol agent in organic farming? So it is Trichoderma species, Trichoderma viridis. It is parasite on soil-borne and foliar plant pathogens. Answer is option B. Then antibody in the serum of a neonate will be, it will be IgG of maternal origin. Answer is option C. Lack of thymus lead to increased infection in humans. Such a condition is called as DiGeorge syndrome. Answer is option C. In trisomic individual, the number of chromosome is 2n plus 1. Answer is option D. Then there are three genes A, B and C. If the recombination frequency between AB is 5% and BC is 10%, calculate the map distance between AB and BC. So naturally, recombination frequency is nothing but map distance expressed in centimorgans. So here AB will be 5 centimeter and BC will be 10 centimorgans, not centimeter, centimorgans. Answer is option B. Then next one, if a disease is Y-linked, which one of the following assumptions are correct? Transferred from male parent to male child. Yes. All the male progenies will be affected. Yes. It is a mitochondrial disease. No. Answer is both A and B. Then inactivation of P53 lead to. So P53, it is a tumor suppressor genes and its inactivation lead to tumor formation. So answer here is option D, that is both A and C. That is formation of tumors and unregulated cell cycle progression. Next one, in which phase of the cell cycle there is duplication of genetic materials, that is DNA division, DNA is getting replicated, that happens in the S phase. Answer is C. Then in eukaryotes, which one of the following is not a second messenger? So second messenger are intracellular molecule released by the cell in response to exposure to extracellular molecules, that is, or else I can say in response to exposure to first messenger. So out of these, that is 1,2-diacylglycerol, inositol 1,4-triphosphate, cyclic AMP, cyclines are not secondary messengers. All the others are secondary messengers. Then during generation of an action potential, depolarization is due to. So depolarization is due to rapid rise in membrane potential. As a result, sodium channels open and large influx of sodium ions take place. So Na plus influx. Answer is option C. Transport of water across plasma membrane is regulated by aquaporins. Answer is option C. The nucleus is not important as the site of protein synthesis. It takes place in the cytoplasm. All the others take place in the nucleus also. Nucleus. The classic experiment on the sequence of organelles that constitute the secretory pathway carried out by George Palade and his colleagues is. So it is rough endoplasmic reticulum, Golgi, secretory vesicles, cell exterior. Answer is option A. The next one. Which of the following enzymes do not occur in the lysosomes? Phosphatase, lipase, protease, polymerase. So it is polymerase which does not occur in the lysosomes. Answer is option D. Secondary water treatment involved. So the secondary stage of treatment, it removes about 85% of the organic matter in sewage by making use of bacteria. The principal secondary treatment technique used for are the trickling filter and activated sludge process. So answer here will be option C, that is removal of dissolved organic compounds by activated sludge or trickling filter. Answer is option C. The next one. The fungal disease that affects the internal organs and spread throughout the body are called. So they are called systemic mycosis. Answer is option B. The staining technique used to stain the metachromatic granules of Corynebacterium. It is Albert stain. Answer is option B. Then which test is used to identify influenza virus? It is A plaque formation. Answer is option D. Then next one. Which strain of fungi is used for the production of streptomycin is? It is Streptomyces species. Answer is C. First genetically engineered and biotechnologically produced vaccine was against hepatitis B, Angerix. It was called Angerix. Recombinant DNA vaccine. Answer is option D. Lyophilization is a technique that involves removal of water for increasing shelf life and also for transport. So it involves freeze drying. Answer is option A. DNA polymerase used in PCR technique is isolated from which of the following microorganism? It is Thermus aquaticus. Answer is B. Non-leguminous plants carry out nitrogen fixation by nitrobacter. Answer is B. Tricarboxylic acid cycle take place in which one of the following? So it take place in the mitochondria. Answer is option A. Lichens are symbiotic association of fungi and algae. Answer is option C. Then antibiotics are synthesized in which phase of the microbial growth? So antibiotics are synthesized in the stationary phase. Answer is B. Next one. The advantage of mycorrhizal association with plant roots. It is improved mineral absorption, protection from pathogens. Answer is option C, both A and B. Then which one of the following does not belong to edible group of mushrooms? It is Amanita phalloides. It is a poisonous Basidiomycetes fungi. So it is not an edible mushroom. Then the cold loving microorganisms are called psychrophiles. Answer is option B. When the virus integrates its genome with the genetic material of host, such a condition is called as prophage. Answer is option A. Then which one of the following is not a macronutrient for microorganism? It is cobalt. Answer is option D. In a microbial culture, a log phase between two lag phases is called dioxic growth. Answer is option B. Then the other name of glycolysis pathway is Embeden-Meyerhof pathway, EMP pathway. Answer is option A. Which of the following is not a eukaryote? Saccharomyces is a eukaryote. It is a fungus. Trichoderma is also a fungus. Salmonella is a bacterium, so it is not a eukaryote. Fusarium is also a fungus. Then tuberculosis is caused by which group of microorganism? It is caused by bacteria. It is called Mycobacterium tuberculosis. Bacterial species involved in the preparation of ethanol. It is Zymomonas mobilis. Answer is option A. The enzyme used in lactic acid fermentation is lactate dehydrogenase. Answer is option B. Then next one. Which one of the following is absent in prokaryotes? So prokaryotes, nucleus is absent. Ribosome, cell membrane, mesosomes are present. A specialized cell of fungal pathogen that infects a host is called Hostorium. Answer is option A. E. coli was first isolated by Escherichia. Answer is option B. In electron microscope, source of electron is from. So it is from mercury lamp as well as tungsten metal. So answer is option C, that is both A and B. Smallpox vaccine was first discovered by Edward Jenner. Option D. Spirulina belong to which of the following group? It is belong to Cyanophyceae. Answer is B. The dengue fever virus is arbovirus. Answer is option C. Which one of the following is not true of fungi? Contain no peptidoglycan, are prokaryotic. They are not prokaryotic, they are eukaryotic. Susceptible to greaseoulfin and have nuclear membrane. Then attenuation of virus means. So attenuated virus is a weakened, less vigorous virus, and attenuated virus may be used to make a vaccine that is capable of stimulating an immune response and creating immunity but not causing illness. So answer will be inactivation of virus. Answer is option B. So thank you for watching my channel. Please do like, share and subscribe. Thank you.